What do fireflies, squid, and algae all have in common? They all have the ability to glow. Now, I know what you're thinking. Of course you've seen fireflies glow. And you saw that one squid glow on that nature documentary one time. But algae? It's true. Some species of algae, or dinoflagellates, are equipped with the biological capabilities to glow. So what are dinoflagellates? Dinoflagellates are single-celled aquatic organisms with two dissimilar flagella, or little tails. Dinoflagellates are protists, meaning they are eukaryotes that don't really fit into the kingdoms of animal, fungi, or plant. They have some cellular similarities to all three, but they don't really fall into any specific category. Think of them as being the etceteras of the eukaryotes. Dinoflagellates are extremely common. There's an estimated over 2,000 different types, of which 1,700 are found in saltwater and the other 300 are found in freshwater. And of the over 2,000 types, only about 70 of them have been found to have the necessary specialized organelles to make them glow. Like mentioned before, dinoflagellate is just a different word for algae. Perhaps the most known kind here in Florida is the one that caused the infamous red tide. The red tide is caused by a toxic type of dinoflagellate called the Crania brevis. In fact, the Crania brevis is just one of many types of dinoflagellates that are found to be toxic to humans and other animals. And these are some examples right here. On the left, you'll see the Serratium tripos, which is just one dinoflagellate that I personally thought looked pretty cool. And on the right, you will see the Nautica scintillans, or sea sparkle, which are very common, especially here in Florida, and do have the ability to bioluminesce. So now, what are the anatomy of the dinoflagellates? This image is an ultrastructure of the typical dinoflagellate cell, which perhaps is redundant to say as dinoflagellates are unicellular. And as mentioned before, there are over 2,000 different types of dinoflagellates, so the anatomy varies species by species. And I will preface, this dinoflagellate in particular does not have the capability to bioluminesce, and it is autotrophic and does photosynthesize. Since generally the dinoflagellates that have the ability to bioluminesce are heterotrophic and gather their food and nutrients through phagocytosis, I'm not really going to touch on the phycoloids or this whole upper section right here. Now first off, the cell is divided into the upper epicone and the bottom hypocone, with the cingulum right about here in the middle. And like mentioned before, dinoflagellates have two dissimilar flagella, the transverse going right through the middle right here, and the longitudinal, and this is that long flagellum right here. The transverse flagellum is found in the cingulum, and the longitudinal flagellum is found in the sulcus, and it runs perpendicularly to the cingulum. So cingulum's right here, transverse flagellum, is found going through here. The transverse flagellum has a helical axonome, which makes it all ribbon-like and all windy, and it makes it have this really cool pattern right here. The cell covering is called the amphysema, and that's this going around the cell right here. The amphysema contains flattened vesicles called alveoli, and it's lined cellulose fragments called the thecal plates. So you'll see the thecal plates right here, right here, right here, they go all around. And their function is to add extra protection and provide the cell with structure, much like you'll see with cell wall in plant cells. Dinoflagellates often have various kinds of extrasomes. For example, this one has trichocysts right about here and right here, and you'll see one up here. And these usually lie perpendicularly to the amphysema and are used for defense and to excrete waste out of the cell. Dinoflagellates have a unique nucleus right here called the dinocharon, with chromosomes that remain permanently condensed through both interphase and mitosis. A large part of the dinoflagellate cell is filled with vacuoles. You can see one here, and here, and here, and right about here. There's many of them. Including, so with the vacuoles, they'll see two specialized pustules called the sac and collecting pustule that are present in most of the heterotrophic marine species. So right here is the collecting pustule, and this one is the sac. Pustules are connected to ducts that open near the base of the flagella and help controlling what comes in and out of the cell. The dinoflagellate mitochondria, which is right here and right here, they have the tubular cristae. And then many of the organelles and dinoflagellates function the same as they do in plant and animal cells. The Golgi apparatus, right here, is in charge of packing and transporting lipids and proteins in the vesicles that are transported through the cell. Kind of like the cell's little personal post office. The ribosomes, which you'll see these little dots right about here, are in charge of translating the genetic codes into amino acids and then building proteins from said amino acids. And then finally, the endoplasmic reticulum, which you will see right about here, just like in animal and plant cells. They aid in the synthesis, folding, modification, and transport of proteins. And now it's time for the really cool stuff. How do the dinoflagellates actually glow? Now, for this explanation, I'm going to use a dinoflagellate that we saw earlier, the Noctulia scitolon, or sea sparkle, as it's commonly called. And sea sparkle is actually found worldwide, including right here in Jupiter. I've seen it firsthand, I think, sometimes if you go to the beach around the summertime and you run in the sand will actually glow behind you. And although I don't have the special tools to determine whether or not this is sea sparkle, there's a couple of reasons I'm led to believe this, as there's quite a bit of sea sparkle found in Pensacola and up in the upper panhandle of Florida. Anyway, getting on with it. In most bioluminescent dinoflagellates, it's actually controlled by an internal biological rhythm, meaning the dinoflagellates only really glow at nighttime, especially the ones that are seen on the surface of the water. Down below, they don't really have the circadian rhythm that the ones on top do. However, it does help. There's luminous chemicals, and they're stored in vesicles called scintillons. The scintillons move into the cytoplasm from the area around the nucleus. However, from my research, I was not able to find how these scintillons are moved there. From what I saw, scientists haven't really figured it out either. And then bioluminescence requires a lot of energy. A single photon of green light requires about 8 ATP molecules. Luciferase, a specialized enzyme, has a cofactor called luciferin, and it forms a highly strained compound when fused with oxygen, using an ATP molecule to help set everything up. When an oxygenated luciferin breaks, it forms carbon dioxide in the process, and it leaves behind a highly excited form that then emits light. However, all of this is dependent on the pH of the water. This is why sea sparkle and other bioluminescent dinoflagellates are only seen under very special conditions. When the luciferin is not bound to oxygen, it is usually bound to a protein called the luciferin binding protein. Very original, I know, or the LBP. And while at a neutral pH of 7 or even 8, the luciferin binding protein stabilizes the luciferin from being oxidized spontaneously. At a pH lower than 7, however, luciferin dissociates from the luciferin binding protein and then associates with the luciferase. This causes the luciferin to become spontaneously oxidized. And again, in the process of being oxidized, luciferin briefly gets excited, and then at which it decays into its ground dormant state, releasing energy in the process of form of light, and also releases carbon dioxide. The light is usually blue or green, as It's believed that the marine animals have evolved to release this blue light because blue light travels farthest and water absorbs other colors quicker. And with sea sparkle, the whole process is a result of mechanical stimulation. And speaking of evolution, you're probably wondering why sea sparkle and other dinoflagellates have evolved to glow. What's the purpose? But why? Dinoflagellates actually have evolved to glow as a result of mechanical stimulation in order to be used as a way of protection. It seems backwards, I know. How could something like a distracting glow help protect them? You would think it would make them more prone to predators. Well, it goes like this. Sea sparkle and other dinoflagellates are at the very, very bottom of the food chain. Their predators include small marine animals like shrimp. This is a bottom feeder shrimp right here. As you can tell, he's see-through. This is important for later. So when the shrimp is swimming around or eating the sea sparkle and other bioluminescent dinoflagellates, they will begin to light up. And when they eat it, the glow will go through their stomach and they will become visible to other predators. So when they glow, the shrimp will either be scared away or it will attract the shrimp's predators, which then the shrimp will either be scared away by the predators or it will be eaten. Either way, it all works out and the shrimp goes away. And although this is unrelated, I do find it interesting how other bioluminescent evolution has come to be. You know, fireflies, for example, they bioluminesce as a way to find mates. Anglerfish have developed a way to gather bioluminescent bacteria and hold it in their little sac on the front of their head as a way to get prey. And yeah, I just find it very interesting. And then these are my sources right here. And thank you so much. This is my project.